Hey there, it's Dr. Guerrero. I was going to go over what you should know about the labs for lab exam 3 that's coming up um, for intro to biology. All right, the first labs that we did were involving the mitochondria and the process that occurs in the mitochondria, which is cellular respiration. Uh, Initially, I wanted you to understand the anatomy of a mitochondria. All of the stuff I'm covering here, you're going to hear me say in lecture. I talk about this in more detail. So if you need more guidance on it, roll into the lectures that are available on the playlist. Okay, but mitochondria, you need to understand the three unique characteristics. Mitochondria have two cell membranes or two plasma membranes due to the endosymbiotic theory. I talk about that in lecture. Mitochondria also have one circular chromosome, which is their own DNA, and they replicate through binary fission. All of these are evidence that a mitochondria at one point was a living organism on its own in the form of a prokaryote, which is a bacteria. Okay. As far as labeling this mitochondria, you need to understand the inner and outer membrane, okay, because it's got two cell membranes, the outer one and then the inside one. Then you also need to understand the term cristae, which is just the folded area, okay, and then matrix. Those are things that you will have to label for the mitochondria. In the cellular respiration lab, we actually had two of them. Half the class did one, half the class did the other. The first lab that half the class did was over aerobic respiration. Aerobic means that it has access to oxygen. So there's breathing, like there's nothing inhibiting oxygen flow. We tested three different types of beans. We tested dry beans that had nothing happening to them. We tested germinating beans, and germinating means to grow. So beans that were growing. And then we tested beans that had been growing, but we made them really cold. Okay. So our goal was to measure the amount of CO2 that they released. So if a cell is going through, or if an organism is going through cellular respiration, it's creating ATP. One of the byproducts is the gas carbon dioxide. So the probes that we used in the lab, which were considered CO2 gas sensors, were measuring the amount of carbon dioxide that was released by the beans. These beans were put into a plastic chamber, which was that plastic little container that we had there. You put the beans in there, and then you put the CO2 gas sensor on top of it, and you hook that up to your Veneer Lab Pro, the computer that would calculate the amount of CO2 that was released. What we saw was that the dry beans, there was no slope because they're dry beans. They're not growing, so they're not using ATP or phosphorylating ATP, so they're not releasing CO2. Okay, so they're not carrying out cellular respiration. Then the germinating beans, the ones that were in the room temperature, we saw that the amount of CO2 continued to increase steadily because those germinating beans were using ATP and phosphorylating it, so regenerating it or recharging it, and then releasing high amounts of CO2 that started to compact. So we saw a little bit at the beginning, but as we left it in there, more and more CO2 built up. Now the cold germinating beans, because they're germinating, they're growing, but we made them cold, it slows down their metabolism. So what we see there is they still have a slope increasing, but it's just not as much CO2 is produced because it's slower, because it's cold. It slowed them down. Okay, so that's that lab. Then we go on to the fermentation lab, which was the second part of the cellular respiration that we did, but fermentation was anaerobic. Anaerobic means no access to oxygen. So this may be taking place in conditions where no oxygen is present, may be taking place when someone's sprinting, swimming, weightlifting. I talk about that in lecture. What we did though in this lab is we took yeast, which is a living single-celled organism, and we gave it three different types of food sources. The first food source was glucose, so straight up sugar, simple sugar like you would get in a candy bar. Then we gave another tube of yeast. We gave it starch, which is like eating heavy breads, right? And then we gave a tube starch with amylase. Starch, again, is heavy, like a large carbohydrate. And we put it with amylase. Amylase is an enzyme, and we know that because it ends in ASE. and enzymes are proteins that speed up reactions. So amylase breaks down starch. So just a recollection, in this lab, we set up fermentation tubes that kind of looked like this, and then they had their little stand. You put your yeast and its food source here, and then you put it in the incubator and it stayed in there. We did it for, I think, 10, 20, and 60, so different time increments. And what we were measuring is the amount of CO2 that was produced, because remember, we're still looking for CO2 for evidence of cellular respiration. The CO2 would be collected here at the tip of the tube, okay? So the longer we left the yeast in there, if it was eating a food source that it liked, it would be creating a lot of energy, and there would be a lot of CO2 left as a byproduct. We found after, because you set up 3 tubes, the tube with the glucose, which is simple sugar, did the best, quickest, okay? So we saw a lot of CO2 there initially. Then the tube with the starch, so that's like eating just bread by itself, like a large carbohydrate, because remember, glucose is also a carbohydrate, but starch is a big carbohydrate where glucose is a really small one. When we fed that yeast starch, it was hard for it to break up, so we saw very little CO2, okay? However, if you were able to run the 60-minute trial, the starch with the amylase also produced quite a bit of CO2, not as quickly as the glucose, because the glucose was already ready to work. It was already a simple sugar. But after some time, when the amylase had to break down the starch, then the starch had become a simple sugar, components of simple sugars that yeast could use to phosphorylate ATP, and as a result, CO2 would start to increase, CO2 levels would start to increase. So over time, the starch with amylase does well, but in a quick situation, it's glucose. Starch by itself is not a good food source because it takes too long for it to be broken down on its own. You do need to know the equation for cellular respiration, know the reactants in the product. So what goes in, what goes out. Be able to write the equation and be able to read it. It is on both the lab and the lecture exam, okay? And I talk about that in the lecture, know reactants and products. Okay, in the cellular respiration lecture, we did that big mitochondria. Go get that. Chloroplast is the another organelle. So we just finished mitochondria. Chloroplast is the next organelle we really focused on. Chloroplast, just like the mitochondria, has 3 unique characteristics. Number 1, it has 2 cell membranes. Number 2, it has 1 circular chromosome. And number 3, it divides using binary fission. I cover all of that in depth in lecture. Because of those 3 characteristics, it gives us evidence that chloroplast once survived as a living organism on its own, a prokaryote, also a bacteria. Okay, so we have that evidence. As far as, and the way those characteristics help us understand the endosymbiotic theory, okay? And I referenced that that's the same theory for the mitochondria. Now, for labeling this chloroplast individually, you know both the inner and the outer membrane, okay? One of these little things, I call them a green pancake in lecture, is a thylakoid. You do need to know that that is a phospholipid bilayer. A stack of thylakoids is called a granum, but if you have multiple stacks, those are called grana, grana, that's plural. So this is 1 and this is more than 1, okay? Then there is the stroma. Stroma is the space in between. It's liquid filled, very much like cytosol that you would see in the cell, but it's inside the chloroplast instead. So stroma. You do need to be able to label all of those on a chloroplast. Also, the cross-section of the leaf. Some of the labs I got to go over this, some of the labs I didn't. There is a model inside the lab, but I just included a picture here just to make sure everybody gets it. We talked about the anatomy, the top of the leaf being where the sunlight hits. And so we wanted the chloroplast to be organized really tightly up against the top of this. so that they can get as much sunlight as possible to carry out as much photosynthesis as possible. This area of the leaf is called the palisade mesophyll. You will have to put that on your lab exam. You will have to identify that. Okay. Then we also talked about the fact that this area down here is very spacious. It is referred to as the spongy mesophyll, as it's got a lot of tubes and tunnels in this open space. This is where gas exchange occurs. And we see these structures down here, these pores that are called stomata, not to be confused with the stroma inside the chloroplast that I talked about on the previous slide. Stomata are the location of gas exchange. So just a quick reminder, CO2 goes into the plant and plants use CO2, carbon dioxide, to carry out photosynthesis, and oxygen is released. So CO2 in and O2 out, okay? Also, in lab, I talked about these stomata being holes, and the reason why they're on the bottom side of the leaf is because when the sun hits the top, it causes the water, which comes up in the roots of the plant, to get sucked up to the top. And sometimes it's so hot that water tries to leave the plant. If those holes were at the top, the water would evaporate. But because those holes are at the bottom, it gives the water an opportunity to drop out back into the soil so the plant can reabsorb it. Okay, so structures that you need to be able to label: palisade mesophyll, spongy mesophyll, stomata. And the last thing to point out is this structure right here, the vein. A vein is used to transport water throughout the plant. The photosynthesis lab that we did, there were two different ones that we did involving the chloroplast. We did the photosynthesis and we did chromatography. Okay, so photosynthesis lab, what we did is before lab, I prepared chloroplast. I remember telling you, hey, I took these spinach leaves and I activated them with light. Then I added some glucose to them, so they were so super happy. And then I turned the blender on and severed everything. I created a chloroplast suspension. I poured half of that chloroplast suspension into a brown bottle, also called an amber colored bottle, to prevent any sunlight from coming in. And that unboiled suspension had chloroplast that were activated by glucose and by light, so they were pretty happy. Then I boiled the other half of that suspension. When I boil it, when you boil something, you change the temperature, increase the heat for an extended period of time. The term is denature. It means I change its shape, so I change its function. So I change the shape of those chloroplasts so they can no longer carry out photosynthesis. Kind of think about it in your mind like if you were boiled, basically you would die, so you could no longer carry out your life functions. Okay, so that was the second substance that was in the back that we tested. The third was DPIP, and I told you that DPIP was a synthetic or lab-made version of the electron carrier used in photosynthesis, which is NADP. Okay, so it actually carries out no photosynthesis on its own. It just transports electrons back and forth. What tools did we use in this lab? We used the cuvées, remember, in the cuvée stand, and then the colorimeter or the colorimeter that measured the wavelengths and the rate of photosynthesis. Okay, so that's what I talked about. Now, what did our outcome or our results reflect? So the DPIP, it asked you, first of all, we did water. Remember, water was for a control. It even called it calibration. Okay, then the DPIP, we know that's not going to carry out any photosynthesis because it doesn't have any chloroplast on its own, DPIP by itself. So we saw the slope of that to be super low. The numbers would have been low on that. Then we do the boiled. Let's talk about the boiled. The boiled was denatured, right? So more than likely, if there was any photosynthesis taking place, it was by accident. Maybe not. Maybe there was some chloroplasts that were super strong and made it through all that heat. But the photosynthesis level was pretty low, even maybe zero, okay? Versus the unboiled, which was stimulated by the light, and it was given some glucose, so it was happy. It's got a lot of sugar. Its photosynthesis rates were probably pretty high in comparison, okay? So just talking about that, referencing those slopes as your activity levels. Then we talked about chromatography. So remember that chromatography is the separation of pigments. Okay, that's the practice of separating pigments. And in order to do this, remember we took a spinach leaf and we assaulted it and got it to leak out its chloroplast pigment at the bottom of the TLC paper, which is thin lined chromatography paper. Then we put some chromatography solvent in it, a very little bit, to allow the pigments to begin to separate. We let it sit while we carried out the photosynthesis, light rate photosynthesis lab. Then at the end of the lab, I came back with you and I said, hey, I want to explain what you should have gotten. What we should have seen is the pigments begin to separate, okay? And the heavier or the less soluble, so if they're less soluble, they didn't move very far. So this first pigment that didn't move far at all compared to the rest was chlorophyll B. Chlorophyll A was a little bit more soluble. Then we have xanthophyll, which is a yellowish pigment. And then carotene traveled the furthest, okay? So that's the results that we should have gotten on that TLC strip. What we tried to calculate is the RF factor. So how far did it travel, its solubility? In order to do that, you measure from the starting point how far that individual pigment traveled, and then you divide it by the total distance pigment traveled. And that's how you get that RF factor. That also helps you fill out your chromatography questions that are due in class through upload, okay? Also, make sure you know the equation for photosynthesis. Just like I asked you to know the equation for cellular respiration, know the reactants, know the products, know them from the molecular formula, like for photosynthesis. It's carbon dioxide and water, and that creates C, that's supposed to be a C, C6H12, okay, and my pen I think just died, H12O6 plus CO2, no, O2, I apologize, and make that you get the sunlight energy, or ATP. It's not writing very neat. Okay, so molecularly, they're written out like this, but also that you can write the words out, and I do that in the lecture. So like carbon dioxide, water, glucose, and oxygen. And I cover that really well in the lecture on photosynthesis. Also, reactants are on the left side of the arrow, products are on the right. The lab that we concluded with was the mitosis and meiosis lab. For mitosis, we looked at two different organisms. We looked at a root tip of an onion plant. So this is a plant cell going through mitosis. And then we looked at a whitefish blastula. A whitefish is an animal, a blastula is an egg. So we're looking at fish eggs. So this was mitosis in an animal cell, okay? For the lab exam, you do have to recognize what each stage looks like. And you have to recognize whether it's in a plant or an animal cell. So plant cells tend to be more square, okay, because they have the cell wall, where animal cells tend to be more oval or circular shape in reference to these diagrams here. These are two ones that I just pulled off of the Google, so you can just Google animal cell mitosis, whitefish blastula mitosis, if you want a different diagram that's better labeled for you to study. I also give you the option to take a picture of them on the wall in lab. Questions that you'll have to answer that aren't identifying pictures are questions about mitosis, the cycle itself. So there is the cell cycle, and mind you, this is in the lecture with a lot of detail. The cell cycle is the life cycle of the cell, okay? The first part of the cell cycle is interphase. Interphase takes place in three parts, G1, S, and G2. Interphase is not part of mitosis. Mitosis is this part right here. And then cytokinesis is the end of the cell cycle. So the cell cycle has interphase, mitosis, and cytokinesis. Mitosis has prophase, metaphase, anaphase, and telophase. Again, I talk about this, I give you a diagram to use inside the lecture. You also need to be able to tell me what happens in those stages. So like a question may say, in what phase of the cell cycle is DNA replicated? And you would need to be able to answer that as S phase. what phase of mitosis do the chromosomes line up on in the middle of the cell? And you would need to know that that's metaphase. And I'm just making those words up in my head as I'm recording right now, but you already know that it may be a little bit different words, but that's what it's going to be asking you, the descriptions of what happens during those each of those phases or stages. Meiosis occurs only in gametes, so sperm and egg cells. I have a whole lecture on this as well, but what we did in lab is mess with these pop beads, and you ultimately simulated the process of meiosis and went through the steps. The reason that meiosis is different than mitosis is because there's crossing over that occurs. Crossing over allows for genetic variation, which is why every sperm ever produced by a male is different and all the eggs produced by female are different because of crossing over. So meiosis is different than mitosis as far as the products and the cells that it actually occurs in. Okay, I also, because I knew I was recording this and you couldn't click on it, here is the hyperlink, the YouTube address for this video right here. This gentleman goes through all the stages of meiosis using the pop beads like we do in lab, okay? You also need to be able to label these pop beads, what they represent, and what the individual beads stand for, and it tells you that in your lab manual. Also be able to identify like a chromatid, sister chromosomes, homologous chromosomes, a bivalent, a tetrad, and know what leads to genetic variation, okay? I hope I covered everything. I'm pretty positive I did since this is round 2. I hope that you study and good luck.